Hello, today I'll be presenting a digital case study of a hypothetical scenario in a clinical nutrition context. In this case study, the client is a 48-year-old female seeking nutrition advice to help with anxiety and mild depression. Additionally, the client has been recently diagnosed with colon cancer and is also seeking some help in that regard. So I'll talk about some nutritional strategies that may help with mental health and colon cancer. In the context of the client's anxiety and depression, neurotransmitters like serotonin and GABA are very important. And of course, there are many other neurotransmitters like dopamine, acetylcholine, and others, glycine. But according to Bolu 2022, serotonin is a neurotransmitter that modulates behavior and neuropsychological processes such as anger, aggression, mood, and even sexuality. The majority of serotonin is produced in the gut, but it cannot pass the blood-brain barrier. However, a study by Lewinsky and others in 2023 suggested that neurotransmitters produced in the gut may impact the brain via the enteric nervous system. The essential amino acid tryptophan can be converted into serotonin in the neurons in the raphe nuclei located in the brainstem. Serotonin production is modulated by serotonin receptors, which can be inhibitory or excitatory. In this diagram, we can see the actual synthesis of serotonin. So tryptophan, which is acquired from the diet, and it's an essential amino acid, can first hydrolyze into 5-HTP by the TPH enzyme, which requires oxygen, NADPH2, BH4, and either iron or copper. Other support nutrients include vitamin B9, B2, C, B3, zinc, and magnesium. This 5-HTP is then decarboxylated by the enzyme AADC into serotonin, with vitamin B6 being a cofactor and vitamin B2 a support nutrient. Moving on to GABA, another super important neurotransmitter, the most important inhibitory neurotransmitter in the central nervous system. And this particular neurotransmitter is modulated by the glutamate GABA glutamine cycle. So within neurons, GABA is produced from glutamate by the GAD enzyme, along with cofactor again, vitamin B6. Support nutrients include zinc, magnesium, and taurine. The GABA produced is then recycled within the neuron. And there's other enzymes involved in this recycling process. And this includes GABA-T and SSADH. GABA-T converts alpha-ketoglutarate generated via the Krebs cycle into glutamate, and GABA into succinic semialdehyde. This enzyme again requires vitamin B6. SSADH converts succinic semialdehyde into succinic acid, which can then be recycled into the Krebs cycle, and this enzyme requires vitamin B3 and B1. So overall, from a nutrition perspective, it is important that the client consumes foods with adequate amounts of nutrients discussed in these precursors and cofactors to support neurotransmitter production. Supplementation may also be required, and some foods are also rich in GABA, and this includes things like fermented foods like sauerkraut, yogurt, kefir, to name a few. Now moving on to the colon cancer piece. So I'll be referring to a review article by Zhang and others, 2020, which defines epigenetics as heritable alterations in gene expression that do not result in permanent changes in the DNA sequence. So they're like switches that can be turned on and off. There are three broad alterations or tags that can facilitate gene expression or silencing. These mechanisms are DNA methylation, histone modifications, and microRNAs. This diagram shows different sections of a DNA sequence. Hypermethylation, or addition of methyl groups in the CpG island promoter region facilitated by enzymes DNA methyltransferases, silence or turn off gene expression. Hypomethylation, or lack of methylation, activates gene expression or turns on gene expression. Within the chromatin region, acetyl groups are added by histone acetyl transferases, or HATs, and erased by histone deacetylases, or HDACs. Acetylation promotes gene expression by weakening the compaction. You can see that in this diagram, that there's more space, and this allows for easier access to transcription factors. Deacetylation silences gene expression by compacting and inhibiting access to transcription factors, and you can see it in the diagram here with a compaction. MicroRNAs are short single-stranded RNAs which target single mRNA messenger RNAs or broadly influence gene expression. All three mechanisms are particularly important in the context of tumor suppressor genes, which suppress cancers, and oncogenes involved in the progression of cancers like colon cancer. According to Cow and others in 2024, environmental factors may drive epigenetic changes leading to the progression of cancers. These include things like aging, sedentary lifestyle, hypertension, cirrhosis, a family history of colon cancer or IBD, excessive alcohol, smoking, obesity, and something that fascinates me, gut microbiome dysbiosis, meaning having the wrong balance of microbes in the gut. There are various nutrients and bioactive compounds that may have positive epigenetic effects when dealing with colon cancer via DNA methylation, histone modifications, and microRNAs. Examples are the one-carbon pathway precursors like folate, B2, B6, B12, methionine, choline, betaine, which promote methylation and the silencing of oncogenes. Selenium is a HDAC inhibitor, promoting gene activation of tumor suppressor genes. Another pertinent example of HDAC inhibitors is butyrate, produced by some species of gut bacteria by fermenting dietary fibers. Introducing these foods may support the client in managing colon cancer from a nutritional perspective. So we've covered serotonin and GABA and how they're synthesized and produced with the cofactors required. We've covered epigenetics and colon cancer and the three mechanisms involved. And we've spoken about nutrients and bioactive compounds that can modulate these said mechanisms. Well, I hope you enjoyed. Thank you very much.